Okay, now before we go on with the importance of meiosis, I want to do that demonstration because it's a whole lot easier for you to understand why it's important if you understand the process completely. And the demonstration will help. So we're starting with, this is a cell. This is the nucleus of a cell, okay? The DNA, you can see, is all just up in there. Now I'm going to take this, okay? This is the nucleus. This here is my cell, okay? All right. The nuclear membrane is going to disappear, and my cell here is in interphase. It is, each one of these is a set of DNA, genetic information, okay? All right, so it is going to copy itself. So it goes from one set to two sets, okay? And they are held together by a centromere. Okay, there we go. And because the cell wants to make sure that things go right and go well, and as part of the cell checks, it stays together. Like they're not attached, but they like to hang out right next to each other, okay? All right, these guys come together and then they double. Okay, and they get held together by a centromere. Okay, one sister chromatid, two sister chromatids, one chromosome. And these guys also stay together. Okay, and they hang out with each other. Okay, they need to stay close to each other so that way they can make sure. Okay, this is our third set of chromosomes that has condensed, replicated, bam, okay, and held together by that centromere. One sister chromatid, the other sister chromatid, so it looks like it could be an X, but it's not. It's two sideways V's, okay? And here we go, the matching copy of the orange stripe. Woo, okay. All right, so it's been copied. We've gone to prophase, right? Now what do we need to do? We need to go to metaphase, all right? So that means that our chromosomes are going to line up on a metaphyseal plate, okay? So I'm picking the center of my cell and I'm lining my chromosomes up on that, okay? So we've got all of the DNA that you need for one cell, and all of the DNA you need for two cells, lined up together, okay? Just like that. So the DNA has copied, it has come together, it is lined up on the metaphyseal plate. Now what happens next? We're in metaphase, we go to, that's right, anaphase, that girl always trying to rip people apart, okay? It is from the centromere, they're pulled apart, okay, from the centromere, they're pulled apart, from the centromere, pulled apart, okay? So now you've got the cells, the two diploid cells starting to come together, okay? Okay, telophase, now cytokinesis. Okay, they are split into two cells, that nuclear envelope is back, and now we have two daughter cells, okay? Both with all of the genetic information that is needed, both diploid cells. Okay, so now let's take one of these cells, and we're gonna make this cell go through meiosis. Okay, so that was mitosis. Now we're gonna go through meiosis. Okay, this point, the nuclear envelope will go away. These cells have the diploid amount of cellular information, okay, and they're all loose and free, all right, woohoo! But they still have to go through PMAT. So they are all happy and joined up, Now they're coming down and they're condensing, okay? And centromere is being added. It is condensing, not copied, not copied. This is prophase 2. There's no copying, okay? So this is just one complete diploid cell's genetic information, okay? And, okay, you have them come together into their sister chromatids, and then prophase, metaphase, you're going to line them up on that metaphyseal plate. It really doesn't matter which order you line them in as long as you've got the sister's chromatids together. Okay, after metaphase, we go to anaphase. That's right, anaphase. We're going to pull them apart and, okay, go. Pull apart, pull apart, pull apart. Now, we've got 2 cells. Okay, 1, 2. Oh, I dropped some of my chromosomes. Oh, there we go. We've got 2 cells, but each one only has half of the genetic information. Okay? These are haploid cells. These are our sex cells. Okay? If you're male, these would be sperm. If you're female, these would be ovum. But you still have one other cell that needs to do the same thing. Okay? So this cell goes through meiosis by prophase 2. We're going to go ahead and leave those on. Okay, they're lining up on the metaphyseal plate. Okay, they're being separated out by Anna. Okay, telekinesis, or yeah, it's still telekinesis, right? Or is that telophase? Lordy Tuesday. All right, so telophase. Okay, they've separated out. And now you have 4 cells. Okay, 4 haploid cells. Okay? All right, now I'm going to do that again. I'm going to run through it again, and we're going to count the chromosomes. Okay? All right. So we would have done this in class, and everybody would have had their hands on these socks in class if we had been together. But since we're not, we've got to do it with these. And no, these are clean socks. They are not dirty socks. They are purchased specifically for this purpose. They've been sewn shut so people can't, put an orange inside them and try to hit people. I don't know. Y'all are crazy. All right. So we have 1 cell that we're starting with, okay? 1 diploid cell. It's already gone through interphase. We're going into prophase. Prophase has them coming together and condensing down with their copies, okay? Okay, so before this is 1, 2, 3 sets of chromosomes. Okay, that's 1, 2, 3, 4, 5, 6. Okay, all right, they come together. Metaphase. That might have confused you. 3 original sets. 1, 2, 3. So 1 set of orange stripe, 1 set of black stripe, 1 set of green stripe. Okay? They've made copies, so they're sister chromatids. Okay? 1, 2, 3, 4, 5, 6. Okay? Metaphyseal plate. Okay, metaphase, anaphase, telophase, cytokinesis. 2 cells. Each cell with 1 set of orange, 1 set of black, 1 set of green. 1 set of orange, 1 set of black, 1 set of green. Okay? Now, one of the cells is going to go through meiosis. So prophase 2. Okay? 1 set of orange, 1 set of green, 1 set of black. In prophase 2, they do not double their DNA. Okay? Okay? In prophase 2, in interphase 2, they do not double their DNA. Okay? All right. Metaphase. Anaphase. Telophase. Cytokinesis. These are now 2 haploid cells with 1 green sock, 1 orange sock, 1 black sock. 1 orange sock, 1 green sock, 1 black sock. Half of the genetic information required for a fully functional self-sustaining cell. Okay? Sex cells, half of that genetic information. Okay? The other cell that was created from mitosis, mitosis 1. Okay? You got your full chromosome black, full chromosome green, full chromosome orange. Okay? They get lined up on that metaphyseal plate, separated out in anaphase, come together in telophase, cytokinesis. Now you've got the cell with 1 copy of the orange, 1 copy of the black, 1 copy of the green. 1 green, 1 black, 1 orange. Now you have 4 separate and unique cells. These are the sex cells. They are haploid. They only have half of the genetic information. They cannot survive, they cannot function as a cell if they do not become fertilized to make a complete cell, which then would be a zygote. Okay? So sperm and egg come together. Now you've got a complete cell. Okay? No sperm, no egg. Sperm, egg. Half the genetic information. Come together. Sexual reproduction. You have a zygote, a diploid cell. Okay? Haploid, diploid. Haploid. Ooh, now I got a trisomy there. All right, haploid, diploid. And yeah, sometimes those mistakes do happen in the separating out. Sometimes you'll have mistakes where extra DNA goes into one cell and another cell is short DNA. Sometimes that will cause the cell to, even if it is fertilized, it would be fertilized with cell that has a lot of information, and it comes together, and that's too much genetic information, because now we have extra. We've got 2 black, 2 green, and 3 oranges. Okay, that is called a trisomy. That can cause genetic diseases and disorders like Down syndrome, Tay-Sachs, Prince Edward syndrome. All of those things are because it didn't split right. But sometimes this not splitting right and creating a fertilized egg would be so messed up there we go, would be so messed up and missing really important required genes that when it comes together, this zygote's like, nope, it doesn't pass the check. And it washes out the cell. and there is a failed pregnancy, okay? But that would be because the genetic split was not going to create a viable organism. And that happens pretty often, okay? This is really neat, interesting, and exciting stuff, and there's some really good reading that you can use on this. Why is that important? Why is meiosis important? I'm back on page 122. Meiosis is important because, let's say this is the mother and this is the father, okay? Now let's say the mother has a very, she has a genetic disorder that she carries in her genes, and it could cause the child to be born with a deformity, okay? But she marries a man who has a dominant gene that does not have that deformity, okay? If they get married and they have babies, those match up, those match up. This would match up, but because the daddy is dominant, you don't see the deformity, okay? His information can cover her information, or vice versa. Also, it leads to some really interesting genetics, where you've got the idea of, okay, you have two genes, two copies, one from the mom, one from the dad. When they come together, which copy is going to be the one that you see, or are you going to see a blended? That's going to be up next. That starts next week, okay, where we talk about blended inheritance and those interesting genetics there. And of course, I will put up lots and lots of videos for you guys, for you to watch and see and understand, and you can do your own Punnett squares, and it'll be great, great fun. If you have any questions about this, don't hesitate to ask, and I will respond one way or another. I will try to get you your answers. Okay, that takes us through the lecture, and then it talks about advantages of sexual reproduction. You need to understand that and read the selective breeding part, because that's pretty interesting, and then do your section review. Okay, if you have the ability to do anything, any demonstration that similar to what I have, that shows me that you've got comprehension and understanding of this content, videotape it, email it to me. I would love to see it. I miss you guys so much. I really, really do. Okay, that's all for today. That's all for this week. Hit me up. Love you guys. Bye.